All right, we are on to Systems and Disease, which consists of Chapter 4, talking about the various body systems, just sort of an introduction to them, and we'll go into each in more detail throughout the rest of the class. This is basically what the rest of the class will be over. And then we have a little section on pathogens and disease. So right up, let's talk about how the body is organized. So we can basically talk about the body in terms of the tissues in the body and then the organs. So tissues are specialized cells of the same type. So a tissue is all the same type of cells that are specialized to perform a specific function. Whereas an organ is one or more tissues. And those tissues come together to perform a more specific function. It can be a different function than each individual's tissues function. For instance, your stomach has muscle tissue. That tissue is responsible for the contraction of the stomach. In addition, it has connective tissues that line the inside, provide layers so that the stomach acid does not damage it, and it has some epithelial tissues, so it's got multiple tissue types. So the organ performs the function of digestion, whereas the muscle tissue moves so that connective tissue protects from the acid, and then you've got some epithelial tissue on top of the muscle tissue. So you've got multiple tissues in there. Each tissue has its own specific function, and when you put the multiple tissues together, you get the organ. There are 4 basic tissue types in the human body, right? We have connective tissue, muscular tissue, nervous tissue, and epithelial tissue. This is our roadmap for the remaining part of Chapter 4. So let's just get into connective tissues. Amazingly enough, there are 4 types of connective tissues that we'll be talking about. Fibrous tissues, cartilage, bone, and blood is also a connective tissue. Let's go through some of the general unifying features of connective tissues, features found in all connective tissues. So one of the features we find in all connective tissue is the ground substance. So the ground substance is non-cellular, and it separates the cells in connective tissue, right? So what you'll have is, think of it as a sheet of ground substance. And then in that sheet, you'll have little cells in various spots, each separated by the sheet. Like if I were to take a piece of paper and poke holes in different parts, the holes would be the cells, the connective tissue cells, and the paper itself would be the ground substance. Now, ground substance is pretty versatile in its consistency. We have fluid ground substance, which we'll probably find in blood, since blood is a fluid. We have a gel-like ground substance, and we even have solid ground substances. Protein fibers are also found in connective tissues. We have collagen fibers. Collagen fibers provide both flexibility and strength. So collagen fibers are both flexible and strong. For instance, in bone, we mentioned osteogenesis imperfecta as a disease, and it's where you have defective collagen, so the bones are extremely brittle. So collagen is a protein fiber, and it provides flexibility and strength. We also have reticular fibers. So reticular fibers are highly branched and mesh-like. They provide a supportive function. So maybe think of it like a layer of cotton, like maybe you'd find in a mattress or a pillow, right? So it's this branched, meshy substance, and it provides support for the cells within it. And then finally, a reticular fiber is more flexible than collagen and less strong than collagen. It's actually the least strong of the 3 protein fibers. The third one is elastic fibers. So elastic fibers, you might guess from the name, are elastic. They have an ability to stretch. Now, they are also stronger than the reticular fibers. So you'll find elastic protein fibers, elastic fibers, anywhere where we need the connective tissue to stretch and deform. Reticular fibers we'll find anywhere we need to provide support. And then collagen fibers we find where we need strength. So next up is a general, again, we're still in general stuff, but we're finally starting our 4 types: fibrous, cartilage, blood, and bone, right? So first up is fibrous connective tissue. So fibrous connective tissue has a type of cell in it called a fibroblast. Fibroblasts are the cells that produce fibrous connective tissue. So they secrete the protein fibers in fibrous connective tissue, and they secrete the ground substance. So fibroblasts, they secrete protein fibers and ground substance. These are the specific tissues that form fibrous, or the specific cells that form fibrous connective tissue. Now, when we take both those fibers and ground substance together, that is what we call the matrix. So the cells within the fibers and ground substance is the matrix, right? It's where your tissues learn kung fu, busting out a timely 18-year-old, 19-year-old reference, 18-year-old reference. Okay, so fibrous connective tissue comes in multiple types. So loose fibrous connective tissues. We have a lot of elastic fibers in loose fibrous connective tissue. These support tissues and organs. They also surround and protect the tissues and organs. So those collagen fibers there provide strength and some flexibility, and then the elastic fibers provide significant flexibility. So we can see in that picture, right, that there are a couple of fibroblasts, 3 pointed out, that are the cells secreting the ground substance and the protein fibers. Then past those, we have those long elastic fibers, and then they're lighter and sort of striped. They're almost transparent in there. I can just use my mouse, actually. So here are the 3 fibroblasts, the nuclei visible, secreting the ground and the fibers. Here's some elastic fibers, right? Long elastic fibers. And then the collagen fibers are these transparent, pinkish, purplish structures in there. You can see they're much thicker than the elastic fibers, and there's a lot of them together. And that's what provides that strength. So it supports tissues and organs. It surrounds and protects them. And at the same time, the elastic fibers allow for expansion of sort of malleable, flexible tissues or organs that can expand and contract. Of course, I have a nice picture here. We can see this loose, fibrous connective tissue. So what you're seeing in here is a little incision into an ankle. So this is our Achilles tendon, right? And we've got the heel just sort of slightly out of view at the bottom. And we've got this incision right here. And so we've got some muscle back there with a layer of connective tissue over it. And then we've got sort of a little fat layer. We've got all this epithelial tissue here. And then this meshy stuff that looks almost like spider webbing from a bad haunted or horror movie or whatever. This is the loose, fibrous connective tissue. So, next up is dense, fibrous connective tissue. From the name, you can obviously tell that it is very dense. So, it is formed from dense layers of collagen. So if we look in the picture, we can see a large number of fibroblasts, and then we have those dark lines pressed together. And this is all thick, dense layers of collagen. Dense fibrous connective tissue is extremely strong. It holds its shape exceptionally well. And so we find it in tendons. Tendons are dense fibrous connective tissue that connect muscle to bone. So we can see in our picture, this is the gastrocnemius or the calf muscle, and it's connected to the Achilles, which is then connected to the heel bone. So that is a tendon. Ligaments connect bone to bone. So in your ankle here, you'd find a number of ligaments around connecting the bone to the bone in order to provide stability in joints. So you'll find ligaments around joints in order to provide stability. Here at the bottom, we have our plantar fascial ligament. Again, you can see that's stabilizing the joint, preventing the foot from completely hyperextending when you set it down. Pretty good example. And both of these are dense fibrous connective tissue made up of those extremely thick layers of collagen. I am pointing all these pictures out because these pictures might very well show up on your exam, and you'd have to recognize the type of connective tissue based on the picture, like the connective tissue in that heel, which was loose fibrous, and the connective tissue in this cellular image, these dense layers of collagen, are dense fibrous connective tissue. So we're picture heavy because I reserve the right to throw them on the exam. Next up is adipose tissue. So adipose tissue is fat storing cells. So the adipose sites in adipose tissue, sites is the suffix that means what now? That's right, cell. So adipose is the fat storing cells. Adipose sites are fat storing cells. So these are relatively dense layers of cells crowded together, and they are the actual cytoplasm with the organelles and everything are actually pretty compressed in there. And most of the cell is taken up by this extremely large vacuole that contains liquid fat. So adipose tissue has a large number of adipose sites. It's rich in adipose sites. And each adipose site has this big, large vacuole with a fat droplet in it. So we can see here's the nucleus. And then if I were to do a little rough outline, this is the cell. You can see the purple cytoplasm makes up very little of the cell. And the grand majority is that big old fat droplet. Adipose tissue is very crowded together, making this thick layer of yellowish fat. So here are some pictures of adipose tissue. So on my left we see a human breast, and this orange stuff in there is adipose tissue. Adipose tissue is very flexible, it's very soft, it provides a lot of support, and a lot of shock absorption. So we see all the milk-producing ducts and tissues suspended in the adipose. So the adipose provides protection and shock absorption for the milk-producing structures. And then over here we have a cutaway, right? So this is a cutaway of someone's leg. And all this yellow stuff on the underside of the skin, this is adipose tissue. So you'll often find connective tissue between layers of other tissues. So the outside is epithelial tissue. Then we have a layer of adipose tissue. Then you'll usually have a layer of like loose fibrous tissue. We can see a little bit of it, this shiny stuff right here and in here. And it all sort of acts to connect and hold tissues together and also reduce the friction when those tissues move against each other. So there we go. Next up is cartilage. So when we talk about cartilage, the big prefix to know here is CHONDR. Chondra. Chondra is the prefix that literally refers to cartilage. Any word beginning with chondra is referring to cartilage. So chondrocyte is a cartilage cell. And we see them right here. This is the ground substance, this very smooth ground substance. Cartilage is characterized by an extremely smooth ground substance. And then we have these little pits with our chondrocytes in them. Right? So, cartilage has a thick collagen matrix. And that is why it has this smooth appearance. The collagen in cartilage gives it a very smooth appearance. A big indicator that you are dealing with cartilage is that it is avascular. These cells in the cartilage, secreting the cartilage, do not have access to blood vessels. This is why tears or rips in cartilage are so hard to heal from, right? Because when it comes time to heal, you need a nutrient supply from the blood. And there is no blood supply to the cartilage. So it's avascular. And there are also no nerves running through it. It's very simple tissue. And then finally, a big indicator is that it is very smooth. So you'll need to be able to recognize this picture as cartilage. And you can recognize that because of the lack of any blood vessels and the very smooth appearance. So we have multiple types of cartilage, and they serve different functions in different parts of the body. First up is the hyaline cartilage. So hyaline cartilage is characterized by very fine fibers, fine layers of collagen. It's extremely smooth, right? You find it in two basic places. One, your nose is made of hyaline cartilage. So you can feel on your skull where the bone stops and the cartilage begins. And this cartilage is hyaline cartilage. You can feel it's reasonably strong. It does not deform very much, and that's because we have fine layers of collagen in there providing that strength. The smoothness and strength is also why you'll find hyaline cartilage lining joints. Joints experience a lot of friction. Anytime you move a joint, there is friction. When you have bone moving against bone, the bone tissue, the friction from the bones, will actually grind the bones and damage them. So, to compensate for that, we have hyaline cartilage lining all of our joints. Provides that smooth, fine layer that almost lubricates the joint. There's actually tissues in there that secrete a liquid that lubricate the joint, but they reduce the friction from the movement of the joint, and they provide shock absorption when the joints move against each other. For instance, when you're walking, especially with your knees, when you put your foot down and you put your weight into it, you can feel your body weight in your knee, and you get compression in there, and the hyaline cartilage both reduces the friction and provides shock absorption. Next up is elastic cartilage. So, elastic cartilage has a significant amount of what kind of connective tissue in it? That's right, elastic connective tissue. So elastic cartilage is characterized by significant amounts of elastic connective tissue, and that provides significant amounts of mobility. And where can you find that? Oh, look at this. Look at this. Oh, look what I'm doing. I'm twisting my ear around and folding it up, right? So your ear or your pinna, P-I-N-N-A. You can find elastic cartilage in the human body making up your pinna, your ear. Lastly, we have fibrocartilage. So fibrocartilage is extremely strong. It's got dense fibrous connective tissue in it. So provides significant amounts of strength. The whole function of fibrocartilage is to resist pressure, which is why the intervertebral discs are made of fibrocartilage. Gravity and movement provide significant amounts of pressure between your vertebrae. So you have those pads, those discs of fibrocartilage providing resistance to the pressure. They also resist tension. So, for instance, if you look at a human pelvis, where it connects up at the front, sort of right below your crotch, right? It's actually two bones. that joint together really tightly. And then there's fibrocartilage lining that joint. They call it the Pubic Symphysis. Symphysis. So naturally, that pubic joint is pulling. There's tension pulling it apart. And so the fibrocartilage resists the tension. So in the intervertebral discs, it resists the pressure. The vertebrae are pushing against each other, and it resists the pressure from gravity in the vertebra. And then in the pubic symphysis, it resists the spreading tension of that pubic, the pubis. Anyway, so that is fibrocartilage. So you'll need to know an example of where you can find each type of cartilage in the human body, in addition to the characteristics of each type of cartilage. All right, we're done with cartilage. Next up is bones. Osteo is the big prefix here. If you see osteo in front of anything, it is bone. So we have osteocytes, and just like with cartilage, we find those osteocytes in pits. So over here in our picture, we can see these dark little holes here. These are the pits in which we find our osteocytes, and they secrete the bone matrix from these little pits. So here are the huge differences between cartilage and bone. One, bone is vascular. You can see in this picture there are columns of blood vessels running the length of bones. And then we also have horizontal blood vessels. And so all these little osteocytes in their pits have a healthy blood supply. So we have this big central cavity, and then in the middle of each of these, let's call them tree rings of bones, we have blood vessels running along the length. And then we have horizontal blood vessels connecting up to each other. And this leads to an extremely healthy blood supply to the osteocytes. So a lot of vascular tissue in there, blood vessels. In addition, this is innervated. This means we have nerves. So we can see right here, nerves in our image running through the bone. So we have both blood vessels and nerves, both of which are lacking in cartilage. Huge difference. So whereas cartilage, its matrix is characterized in large part by collagen, with bone, the matrix is characterized by two basic structures that result in the mineralized matrix. First up, the reason it's mineralized are calcium salts. So these calcium salts provide rigidity to the bone. It allows the bone to maintain a rigid shape. But that rigid shape of the bone is brittle on its own. So in addition to the calcium salts, which is why we say our bones have calcium in them, we also have significant amounts of collagen. And that collagen is providing strength to the bone so that it is no longer brittle. And then we also have elasticity so that the bone can resist vertical pressure and horizontal pressure, right? If you have a horizontal force against a bone and there's no collagen, it will snap. Whereas when you have that healthy collagen in there, it can resist that force. So, it has a mineralized matrix consisting of calcium salts providing rigidity and collagen providing much needed strength and elasticity. So let's talk a little bit more about bone. There are actually different types of bone in the human body. First up, we'll talk about compact bone. So compact bone is the strongest type of bone in the body, right? So it's characterized by rings. So if we look at compact bone here in our figure right here, we have what looks like multiple little tree rings, right? These tree rings are around blood vessels and nerves, and then we have little rings of bone cells in pits, and they spread outward. So these rings provide strength. You have a large number of these little rings densely packed next to each other. You have a lot of strength. So it is dense and it is strong. So we can see right here in this picture, it is what is giving strength to long bones in your body. This is a femur right here. It connects to your pelvis up here. And then this is the surface that connects to your knee, following the mouse. So we can take a little cross-section of the femur and find that the shaft has a lot of compact bone to provide it that strength it needs to resist gravity and shock when moving. So it's the support structure of the long bones. It provides the grand majority of support for long bones. All right. Contrasting compact bone is spongy bone. Spongy bone is really irregularly shaped, whereas compact bone has those nice, densely packed rings. Spongy bone is irregular, consisting of little spars and spikes and rods. So if we look off, our cross-section has a small amount of the center cavity of a long bone, the bone marrow cavity, right? And so if we look at the edge here, this is our spongy bone. You can see it's got these little rods and spars connecting to each other. Provides a lot of open space. That's important because that cavity has to contain tissue for manufacturing red blood cells and immune cells. So it's the bone marrow where we find this spongy bone, providing all that open space for the marrow cells. And as I said, with those hematopoietic stem cells, they produce red blood cells, they produce various types of immune cells, and they produce platelets. And they're found in the cavity, the center cavity of long bones. So that spongy bone serves to provide a lot of space for them, while still providing support, because the spongy bone lines the cavity there. So we get a lot of blood vessel supply, as you might imagine, because the marrow is producing your red blood cells. So lots of open space for marrow tissue to make your red blood cells, white blood cells, and platelets. So spongy bone lines cavities, whereas compact bones are these dense rings that provide strength and support to long bones. And we're through blood. That means we are getting along through our connective tissue. We're going pretty well. We're already at slide 17 of 41. Stay with me, folks. Okay, so the big thing about blood is that it is a fluid connective tissue. So it is still a connective tissue, but it is a fluid, which means we have a liquid matrix. The blood plasma is the liquid matrix of the blood connective tissue. Blood serves extremely important functions. It delivers signals like hormones, it delivers oxygen, and it delivers nutrients to the entire body. Signals like hormones, oxygen, and nutrients. It delivers those three throughout the body. At the same time, it removes waste produced by the cells and takes it to the parts of the body where the waste is expelled. So it delivers signals, oxygen, and nutrients, and it removes waste. So we call the different types of cells in the blood the formed elements. So when you see formed elements, think blood cells. Obviously, we have red blood cells, but we can't say red blood cell. This is science, anatomy. We need a fancy term. So that term is erythrocytes. Erythrocyte is a red blood cell. It is the most numerous formed element in the blood because it's carrying the oxygen. You need a lot of oxygen to run a body. Next up are the leukocytes. Leukocytes are immune cells, what we refer to as white blood cells. So remember when we talked about cancer, just to bring it back to that depressing subject, leukemia was a cancer of the white blood cells, so leukocytes are the white blood cells. And they are the various types of leukocytes in your body make up the grand majority of your immune system. Finally, the third formed element are the thrombocytes. Thrombocytes are very small compared to the cellular formed elements. What we're getting at here with thrombocytes is the reason why these are called formed elements. So when that hematopoietic stem cell goes to make thrombocytes, it turns into a type of cell that produces the thrombocytes, the platelets. Now, the platelets are not a type of cell on their own. Rather, they are fragments of their mother cell. And their whole function is to plug up damage to blood vessels. So, thrombocytes are actually cell fragments. So, this is literally why we call them formed elements. We'd call them the different types of cells in the blood if platelets were actually functional cells, but they are cell fragments. And their whole job is to plug up damaged vessels, and that is called clotting. So the function of the thrombocyte is to clot the blood. Next up in our journey through connective tissue, muscular tissue. Welcome to the gun show. Oh yeah. Oh yeah. Anyway, so muscular tissue's whole job is movement, right? Its whole function is movement. It allows things to move by contracting and relaxing. When a muscle cell contracts, it shortens. When it relaxes, it lengthens. So for the gun show, right, it contracts my bicep. In fact, if I put my arm on it, I can feel it contract. I can feel it shorten up, right? So that's contracting, and they shorten up, and when they shorten up, they, pull the bones. They pull the joint to allow for movement. And when I relax, it allows the joint to extend. So, flexion, extension, contraction, relaxation. That is the whole deal with muscle tissue. All right, so the fiber in this case refers to an individual muscle cell. So on an exam, if I say muscle fiber, a muscle fiber is a muscle cell. Reason we call it a muscle fiber is because a muscle cell is extremely long, right? Think of it sort of like a thread or a tube that's very long. And so it looks almost like a string. And that's why they call it a fiber. And it can contract and relax. So a single muscle cell is a muscle fiber. We have 3 different types of muscular tissue: skeletal muscle, smooth muscle, and cardiac muscle. So let's get to each in kind, and we'll also see the pictures, which will allow you to identify the type of muscle pictured on the exam. I'm giving a couple of hints here and there, as you might have noticed. Okay, so the big thing about skeletal muscle is that it is under voluntary control. You control it with your mind, right? So if I want to move my finger around and point, I think about doing it, right? It's under my control. That is voluntary. Now, when you look at this picture, you can see that these fibers, right? So this is a muscle fiber. You can see it's long. You also see that it has all these dense stripes. These stripes are what allow the muscle cells to contract so much. So between every stripe, we can pull it and shorten it. And so you have all those stripes, which means we can shorten it a lot. So all those bands, those stripes, are called striations. Muscle tissue is striated. That's a big visual indicator that you are looking at skeletal muscle tissue. So we can see how skeletal muscle is arranged in this lower picture here, right? So we have bundles of bundles of bundles. So right here is a single skeletal muscle fiber, right? So you can see that there appears to be lots of little rods packed in there. Those are the protein filaments that allow the contraction of the muscle. So then we bundle these together into this bundle, and then we have a whole bunch of these bundles together wrapped around by a layer of muscle, protecting the bundles and holding the shape of the bundles. And this makes up the whole actual muscle. So a muscle fiber, or a muscle, is not a whole bunch of muscle fibers. It's bundles of muscle fibers inside muscle, a connective tissue holding it together, and then bundles of bundles all wrapped up. So that is skeletal muscle. So it allows movement. It moves your bones. So there we go. On an exam, you'll need to know that it's under voluntary control. Your mind controls it. It is striated, as we see in the picture, striped or banded, and it allows movement of your bones. You ain't going anywhere without skeletal muscle. Next up is smooth muscle. Smooth muscle is different. It is, for almost the entirety of it, involuntary. You do not control it with your conscious mind. You cannot go, stomach, Contract now. Relax, contract, relax, contract. You're not in control of your muscle doing that. As you might have guessed, the muscle tissue around the stomach is smooth muscle. So these muscle cells are spindle-shaped. They're little spindles. Think of them as sort of a swelled-up center that come to little points on either end. Little spindles. Their whole job is to, so there's the spindle shapes. Each smooth muscle fiber is a little spindle shape. And its whole job is to surround hollow organs. It surrounds the digestive organs. It surrounds your excretory organs, your organs that push stuff out. It even lines the walls of your blood vessels. And its whole job is to push substances. Your esophagus, you swallow. Oh, hey. Ow. Some voluntary muscle action with my masseter. Transfer to the esophagus. I can feel it pushed down. I use my voluntary tongue to push it back to my throat. Once it reaches the correct spot, my esophagus takes over and pushes it down to my stomach. Pushes substances through a hollow organ. My stomach will move around and contract and mix things up with the acid and then push it into my intestines. As digestion occurs in the intestines, it will be pushed through the small intestine, pushed to the large intestine, where we turn that substance after digestion into your poop. And then your intestines transfer it to your rectum. Rectum damn near killed them. Hey, I'm hip and with it. Anyway, from your rectum, it gets pushed out. You'll notice that you have some sphincter action under control. Actually, two sphincters there, one that is involuntary smooth muscle and one that is voluntary under your control, which is why you can hold it. That was an interesting little aside for pooping, but, you know, I gotta do me. Oh, big thing. Back on smooth muscle. Look at this picture. What do we not see? We see no stripes, no bands, no striations. If I am showing you muscle, if I indicate that this is a picture of muscle tissue, right, I'll indicate that it's muscle tissue because it looks kind of like the cartilage as well. But if I deliberately say this is muscle tissue, you will look and you will see, oh, look at that. No bands, no stripes. That's smooth muscle. Because cardiac muscle is also striped. It has all those bands. Okay, so cardiac muscle is involuntary. That's right, because you cannot go, heart, stop beating. Okay, heart, beat faster. Nope, not under conscious control. That shit's involuntary. So, cardiac muscle is kind of fascinating because it is involuntary. It is striated. So it has all those bands. And it has a unique to cardiac muscle feature. Intercalated discs. Intercalated discs connect bundles of cardiac muscle fibers. So you'll have this whole line of cardiac muscle fibers, and they end here. This whole line of muscle fibers, and right here we have a thick intercalated disc. This allows signals to pass from this bundle of fibers to this bundle of fibers extremely efficiently. This is because the signal for your cardiac muscle to contract passes through like a wave. And then those signals from there pass through like a wave. And so it allows those waves of signals to pass through the muscle very quickly so that you can have this one contract, and run through the heart so that it pumps all coordinated like. So we have those intercalated discs, these thick barriers between the cells that allow those signals to easily pass along. So if I show you a picture and ask what type of muscle cell this is, you'll see that there's bands here, and you might go, oh, well, it could be skeletal muscle. Then you go, oh, wait, there's an intercalated disc. This is cardiac muscle, baby. On to nervous tissue. We're getting there, folks. We're getting there. So, nervous tissue. Nervous tissue is characterized by a mix of different types of cells. We have a type of cell called the nerve, which is the big characteristic cell of the nervous tissue, but we also have a bunch of little accessory cells to help them out. So first, let's talk about the, unique cell to the nervous tissue, the neuron. So the neuron is composed of 3 parts to the cell. We have the dendrites. When you look at a nerve cell, you will see a bunch of tree branch looking things coming off of it. These tree branches are the dendrites. They connect up to the neighboring nerves. Then we have the big, thick cell body. You know it's the cell body because the nucleus is there. So, if you put soma down, that's the technical term. I'll call that correct, but really you only need to know cell body. And then we have a big old tail coming off the end of it. This long tail is called the axon. And the axon, at the end, does a number of things based on the type of neuron, but for the most part, it branches out into multiple little terminal ends. And each one of these little terminal ends connects up to a dendrite. So we have a big, long highway for nerve impulses to travel. So if I have a nerve cell over here with its little dendrites, a signal comes in, and the dendrites conduct that signal to the cell body, and then it's conducted along the tail, and then it comes down the tail, and we get a little connection here, and a little signal passes along there and says, okay, dendrite, fire! And and the signal passes down here along the cell body, down the axon, and then connects up to the next dendrite. Right? So, big, long wave. Okay. We call neurons by different names depending on where they are in the body. So, we use the term neuron in the central nervous system. Central, C, nervous, in system, S. Central nervous system. This is the brain and the spinal cord. So, your brain up here and the spinal cord running through the center of your vertebra. All right? So, any nerve cell, any neuron in there is literally called a neuron. Outside the central nervous system, we call them nerves. So, PNS is peripheral nervous system. All the nerves coming off of the spinal cord and the brain running throughout your body. So, the brain only has nerves that run throughout your face here. So they're called the cranial nerves, cranial for your skull. So your optic nerve connects up to your brain. The most numerous of your nerves are throughout the rest of your body. So those nerves connect up to your muscles, and the voluntary signal conducts through your neurons in your brain, through the neurons in your spinal cord, out to the nerves in your body, and they go out to the muscle and say, contract! And then when the signal stops, the muscle relaxes. So, they are very large cells. The femoral nerve is quite visible. It's a very thick bundle of axons. So this is the neat part, is that the soma and the dendrites for your nerves in your body are all sort of hanging out up by the spinal cord. And then the nerves that run through your body are big bundles of just axons, which is cool. So like your femoral nerve running through your leg is this very thick nerve. It can be like a centimeter thick, highly visible. Pretty cool. And their whole function, the function of the nerves is to conduct signals. They'll send signals from the nervous system out to the body. And they also send signals from the body to the nervous system. For instance, when my stomach is all empty, signals from my body go to my nervous system and say, I'm hungry. And then I just eat. In addition, touch. Right? Touch and pressure. Those are signals being sent from my body to my nervous system. Whereas contract, that is a signal sent from my nervous system to my body. Pretty cool. And then lastly, we have the neuroglia. So if we look in this picture right over here, the neuroglia are accessory cells. They are not neurons. So here we see these oligodendrocytes. You definitely don't have to know that name or for right now their function, but they're wrapping around that axon, providing a little fatty layer that actually allows signals to travel much faster down the axon. Then we have these little astrocytes. Astrocytes are sort of the bouncers because you have a blood-brain barrier. So any blood vessels heading into the brain are surrounded by astrocytes. So anything coming from the blood first has to go through an astrocyte before it ever gets to a neuron. It provides a layer of protection. Kind of annoying in medicine, right? Because Parkinson's disease is where some of the glial cells that secrete dopamine stop functioning. And if we could just give someone an injection of dopamine, it'd solve the whole problem. But that blood-brain barrier is there, so you can't get the dopamine across to the neurons where it's needed. It sucks. So, astrocytes also help firm up the connections between neurons, so between axons and dendrites. Neuroglial cells. They do not carry impulses. They don't carry those signals to through the brain and the spinal cord out to the body, or from the body to the spinal cord and the brain. They're not carrying signals. They're not carrying information. They're accessory cells. They support the nervous cells, and they nourish the nervous cells. So I like this next slide. It's an excellent picture showing the connections between neurons. So here we go. Here's the axon of a previous neuron. Here's the connection where the axon hooks up with a dendrite. Signal comes down. Oh, hello, dendrite. Transfers through to the cell body. From there, transfers to the axon, comes over to the next connection to a dendrite, which transfers the signal to the cell body, onto the axon, and so on. So right down here is a really cool picture. Very colorful. Kind of looks like an impressionist painting of balloons. But these are colored neurons forming different connected pathways. And all these stringy fibers of rainbow colors are axons moving throughout, forming connections. So when we talk about your brain being made up of neurons, there are literally more neurons in your brain than stars that are calculated to be in the universe. Carl Sagan called your brain an enchanted universe, a universe within your brain, which is a pretty amazing descriptor. So there's another little artist's depiction, just showing you how interconnected these things are. And even this artist's depiction had to provide empty space for you to be able to just comprehend the image. So it's pretty amazing the amount of neurons and connections in your brain. But they have to be there in order to allow you to process the complicated information that they need to. All right, so these are nerves. So here we have the skull containing the brain and the spinal cord. These are neurons, and we can see connected to the spinal cord are nerves. And here's the sciatic nerve, and it transitions to femoral nerve, sort of named based on where it is. And if we actually take a look, we can see just how visible this is. We have a ruler down here showing centimeters, and we can see that this is like half a centimeter thick, and it gets darn near a centimeter thick further in. These nerves are large, extremely large bundles of axons. So, very cool. Then here we have a nice image of the glial cells protecting your brain. So we can see all these little oligodendrocytes connected up to axons here. Then we have these little astrocytes along here, and they're just providing support and nourishment. And you can see here, here's a blood vessel. Then we have an astrocyte connected to the blood vessel, creating a barrier. So this isn't the best image to imply the barrier, but they're actually a very dense layer of astrocytes around there, providing a tight barrier. In fact, some of the most dangerous pathogens there are pathogens whose toxins weaken the blood-brain barrier. Okay, so past the pictures, onto the continuation of nervous tissue. Let's talk about the functions. I said that they carry information. So what does this information term mean? Well, it means that these nervous cells communicate. Neurons communicate with one another. They send signals, right? So signals from the brain out to the body, and from the body to the brain, and within the brain, and within the spinal cord. And we process information, right? When I have those little nerves in there, the nerve cells that detect pressure, the amount they fire is based on the amount of pressure. So more pressure, more firing. And the rate of that fire is sent to that brain, and the brain interprets that to pass on or perceive the sensation of pressure. At the same time, it allows you to control your body, both voluntary and involuntary control. Most of it is from your brain, your nervous system. And as I just mentioned with pressure, it allows for sensation. Okay, on to epithelial tissue. So we've hit connective tissue. We've hit nervous tissue. So now we're on to, we hit muscle tissue. We're on to epithelial tissue, fourth of the types of tissue. And then it's just pathogens, and we're done. We're at slide 28 of 41. If you want to take a break, hit pause. Maybe fast forward about 20 seconds because Professor Jonathan Heckmann needs to eat. Pepsi. Refreshing. Now I need to call them and get money. Okay, the final type of tissue is epithelial tissue. Epithelial tissue covers the outside of your body. It is the interface between your body and the external world. It provides the covering. When you think epithelial tissue, you probably think skin. At the same time, epithelial tissue lines body cavities. So, right, you can hear that hollow sound. There's a body cavity in there where my lungs are. Below there, there's the body cavity where my intestines and stomach are. And the body cavity has a lining of epithelial tissue between the walls of the cavity and the organs inside. We can sort of separate epithelial tissue into two basic types. We have simple epithelial tissue. Simple epithelial tissue is one layer of cells. That's all it takes to be called simple. Stratified, like your skin, is multiple layers of cells. You can think of the top layer as where the environmental interface is, whether it's, the actual body cavity or the outside environment. Let's continue with skin. So this might be the top layer. And then we go down multiple layers. And then at the bottom is what we call the basement membrane. So the basement membrane is where we connect our epithelial tissue to the other tissue in the body, connective tissue that then connects to other tissues. So the basement membrane is the bottom where we anchor to the rest of the body. So stratified epithelial tissue, layers. Think of your skin. Top layer or outermost layer, environmental interface. Move multiple layers down until you get to the bottom, and that's the basement membrane where it connects up to the rest of the body. So hopefully not awful. There are three basic epithelial cell shapes that we'll mention. The first is squamous. Squamous epithelial cells, or squamous epithelium, is flattened cells that are irregularly shaped. So the cells on the outer layer of your skin are squamous epithelium. The cells inside your cheek, right, the lining of your cheeks here, that is squamous epithelium. And again, it can be simple, one layer, or stratified, multiple layers with a basement membrane. Next up is cuboidal. You'll never guess why it's called cuboidal. Oh, it's kind of cube-shaped. Look, all the sides seem vaguely cube-shaped. So, again, this you find in the glands. So, like your sweat glands, you'll have cuboidal epithelium lining them. And you can have simple one layer or stratified multiple layers of cuboidal epithelium. And finally, we have columnar epithelium. So our examples of where we find columnar epithelium is lining the digestive tract and lining the uterus. So here's this blue picture is my favorite of columnar epithelial cells. And then you have this dense layer of cilia topside here. And these move the fluid in the ovaries and the egg down the fallopian tubes towards the uterus. So there you go. This is columnar is interesting. So we know it's columnar because, well, it's longer than it is wide. Columns. But columnar epithelium is not just simple and stratified. Rather, we have what's called pseudo-stratified. Pseudo-stratified columnar epithelium is basically, if you look at this picture, we do not have multiple layers, but we sort of have kind of sort of in between layers. So it's not so much cells stacked on top of each other like with our stratified cuboidal, but some cells interfacing with the top. Some of them go all the way to the bottom, but other cells interface with the bottom and don't go all the way to the top. So it's not stacked, so it's not perfectly stratified, but there are layers of a sort, so it's pseudo-stratified. And we can actually see that here in our picture of our uterine lining columnar cells. So we can see that some of them are sort of smaller and compressed. Right around here we can see, I'm leaning, but you can see right over here that it doesn't quite, this one right here may not quite reach the top. So pseudo-stratified. And then here are some other ciliated cells. So the whole idea is columnar cells line the digestive tract, they line the uterus and the fallopian tubes. So in the digestive tract, their big function is absorption of nutrients. And as I mentioned, in the uterus and the fallopian tubes, they're ciliated and they push the egg along the fallopian tube into the uterus. So there you go. Squamous, flattened, irregular cells. Cuboidal, kind of cube-like. They look about the same width as they are high, and they line the glands. The squamous epithelium, I gave the example of the top layers of your skin and your cheek lining. Their function is for protection. You have multiple layers of stratified squamous epithelium there to provide you protection. Also, materials can diffuse across your squamous epithelium. So, stuff can diffuse across my cheek lining. Water pathogens can cross the cheek lining. So They are also found in some organs for filtration. Multiple functions there. But probably the easiest one to remember is protection. Think of your skin. So, cuboidal in the glands, columnar lining the digestive tract, absorbing your nutrients, lining the fallopian tubes and uterus to push eggs along with their cilia. That is our chapter 4. So now it's on to our bit about pathology. Slide 30 of 41, 11 to go. So here we go. Pathology, the word, refers to the study of disease. Disease at its most basic refers to disruption of homeostasis. So if we remember, homeostasis is the maintenance of internal body conditions. A disease disrupts homeostasis. When we refer to a disease, we're generally referring to a set of signs and symptoms that occur together. Signs and symptoms have very specific definitions. So a sign of a disease is something that you can measure objectively. It's an objective, measured feature. Body temperature. We can take a thermometer and measure your body temperature. White cell count. These are all objective, measurable signs. Symptoms are subjective. Objective is measured by the doctor. Subjective is reported by the patient. I'm tired. I'm woozy. I have a general sense of malaise. One of my favorite genuine medical symptoms is called doom. Doom is a symptom wherein you feel like you or the entire world is doomed and death is imminent. Death, destruction is imminent. It is called doom. It's often associated with diseases or pathogens that affect your brain or your heart. So, anyway, signs, objective, measured by your doctor. Symptoms, subjective, felt, reported by the patient. So, I'm tired. Doc, I'm tired all the time. Okay, so let's talk about sort of the two different ways you can experience a disease. An acute disease, or if you're unlucky, the acute phase of a disease, is sudden and it is short. So think about a 24-hour flu. An acute disease will often have a greater severity. So you'll often find the highest severity, highest sort of danger in the acute phase or an acute disease. In general, the flu is an acute disease. Sudden onset, short duration, can be very severe. We can contrast this with a chronic disease. Chronic diseases generally have a gradual onset. We can see our chart of increasing severity, gradually increasing over time. Chronic diseases are often long-term diseases, so they last much longer than an acute disease. Some of them will last your entire lifetime, which blows. So, for instance, arthritis is a chronic disease. The flu is an acute disease. Okay. Infection has a very specific term. Is a very specific term with a specific definition. An infection is caused by microbes multiplying within you where they shouldn't be. So, some microbes, microbes is microscopic things. So, parasites, bacteria, viruses, these are microbes. They cause an infection when they get in you or on you, and they multiply. So, the more they multiply, the more they disrupt your homeostasis. So, I make the distinction of microbes being where they're not supposed to be because a large part of every person is the normal flora. Normal flora is a term that refers to the microbes that naturally live on you and in you. Bacteria are part of your normal flora. Fungus, single-celled fungus like yeasts, they're a natural part of your body. Viruses, believe it or not, are part of your normal flora. So, normal flora are characterized as being harmless or even beneficial. So, for instance, there are some bacteria on your skin that produce a chemical that is poisonous to other bacteria. Bacteriocins is what they're called. You don't have to remember that. Anyway, they produce a toxin that kills other bacteria. This is great because often the first line of defense that a pathogen has to get past is your skin. So your normal flora on your skin, there are bacteria there that will secrete toxins to protect you from pathogens. Bam! Beneficial right there. Competition is a great way that they help out. So there are E. coli living in your intestines in very large numbers. So they occupy the tissue lining your intestines. Think of it as this big carpet of E. coli, like E. coli apartments everywhere. Because they have apartments everywhere, something that causes diarrhea would first have to get past the E. coli before it could get to the intestines themselves and start causing you problems. So if you have a healthy intestinal flora, you're much less susceptible to diarrhea-causing pathogens. So they can help you through competition. They can help you by being toxic. They can directly inhibit pathogens. And they can just cause no effect and hang. So I want to make this big distinction because while an infection is a proliferation of microbes on you or in you, you have to really consider these as microbes that are either not part of your normal flora or microbes that are in the wrong place. So, for instance, if you get a burn or a deep cut, some of the microbes that are part of your skin flora can get into your blood, and they can cause infection once they get below the skin. So, as long as your normal flora is where it's supposed to be, it's harmless or beneficial. That's why I always tell my students, antibacterial everything is just bad. Antibacterial hand soap is practically useless. The sanitizers in hand soap need time to work. You will notice most people throw some antibacterial hand soap on, wash, rinse, done. There was no time for that antibacterial substance to function on there. You basically did nothing. Furthermore, because it's antibacterial hand soap and you thought you were killing everything off, you probably washed your hands for a shorter amount of time. Soap works by binding up all the crap on your skin and rinsing it away in the water. You want to be practicing sterile technique. You want to be doing good hand washing habits. Run through your ABCs in your head all the way through to next time won't you sing with me, and scrub for that amount of time. That gives you all the time you need for soap to function. And that's still not enough time for antibacterial hand soap to function. Antibacterial hand soap is a waste of money at best, and at worst, you may be putting the bacteria on your skin through a little gauntlet where only the resistant ones survive. So, at best you're wasting money. At worst, you're causing bacterial resistance. Am I saying don't ever use antibacterial products? No. Like if you've had raw chicken on your counter, maybe use an antibacterial substance when you clean that counter off. So, it's generally a lot easier when you wipe down that counter. You'll still have that sterilant, that antibacterial substance left behind, and as it dries, it gives it time to work. So that's fine. I've seen antibacterial body wash before. That is awful and worthless. Hell, Listerine advertises itself as antibacterial. It's frickin' liquor. The alcohol is what's antibacterial about it, and you're just killing a lot of normal flora, and you don't use it long enough to really help, but you do use it long enough to sort of damage the enamel of your teeth, so just brush your teeth. So yeah, antibacterial everything is bad. Respect your normal flora. Love your normal flora. Okay, so let's talk about some common pathogens. 8 slides to go, folks. We're in the home stretch. First up is the bacteria. Bacteria are prokaryotes. Remember, we talked about prokaryotes versus eukaryotes. So bacteria belong to domain bacteria or eubacteria, depending on who's referring to it. Bacteria have a cell wall. It is not a cellulose cell wall. It's a different kind of cell wall. It's not a chitinous cell wall like in fungus. Not cellulose like plants. Not chitin like fungus. You don't need to memorize what type of cell wall it is. Also, there's sort of different types of or thicknesses of cell walls among the different types of bacteria. So really just remember that it's not a eukaryote cell wall. It's a bacterial cell wall. For movement, if they have them, bacteria only have flagella. They do not have cilia. Now, being prokaryotes, all their DNA is sort of chilling out in the center of the cell. There's no organelles, so there's no nucleus. It's just a circular genome chilling out in the cell. Now, here's a very cool thing. In addition to the bacteria's circular genome that tells everything what to do, when to do it, we also have plasmids. A plasmid is extra DNA. So it is not part of the genome. So here we can see our big purple circular genome in the middle of our bacteria. And then over here in a lighter shade is a small circular plasmid. So plasmids are circular DNA because prokaryotes are always circular DNA. These are not part of the bacteria's genome. They're extra. They can carry what we call novel genes or new genes. And what I'm getting at is genes that the bacteria's genome don't got. These are dangerous. Plasmids can code for resistance. antibiotic resistance. So if you've heard of MRSA, methicillin-resistant Staphylococcus aureus. Staphylococcus aureus is a bacterium. Methicillin is an antibacterial, and it's methicillin resistance. That resistance gene that allows it to not be killed by methicillin is carried on a plasmid. Plasmids can also allow a harmless bacteria to produce toxins. So there's a type of E. coli that can cause food poisoning. Gosh, I'm trying to remember the name. I think it's like E. coli 0157H7, but I would not take me at my word for that. I am, you know what? I think I can actually Google this without stopping the recording. E. coli pathogen 0157. Let's see if that's right. Oh, 0157H7. Damn, memory, you're doing me right. Okay, so that is an E. coli that picked up a toxin gene, and I believe it's on the plasmid. So what's dangerous about this is that plasmids can be traded between bacteria. And bacteria aren't really like animals and plants, where each one mates within its own species, right? Bacteria can basically form a bridge between really any two bacteria. If one bacteria has the genes to make a bridge, and one bacteria has the genes to, hook up with the bridge, then they can come together and trade plasmid information. It's thought that the first antibiotic resistance was likely developed in intestinal normal flora because, from the 40s through the 90s, every time someone walked into a doctor and said, "Doc, I'm feeling," the doctor was like, yeah, here's an antibiotic, take it. And so we had huge overuse of antibiotics. At the same time, a lot of people weren't using their antibiotics correctly. My ex-wife was one of these people where they would get antibiotics for their bacterial infection, and they would take the antibiotics until they feel better. And then they would set them aside and go, well, now I have these in case I get it again. I'm a genius! Saving money! But the full course is there to annihilate the bacteria completely. If you do not use the full course of antibiotics, you're not guaranteeing that you have killed off all the bacteria. And when you kill off most of them, but not all of them, the ones that are left have the potential to have mutated and developed resistance. It's bad. It's very bad. So, practice good antibiotic use, people. Okay, let's talk about the different ways bacteria can be pathogenic. So, exotoxins. Exo means what? Where do you find an exoskeleton on a bug? The inside or the outside? Huh? That's right, the outside. Exotoxins are secreted. They're made by the bacteria and then squirted out into the environment. Tetanus produces tetanus toxin. That's an exotoxin. Cholera produces a toxin. Exotoxins, generally, they are not active very long in the body. So the disease or the infection is sustained by the bacteria continuing to pump out more exotoxins. As long as the bacteria is pumping out exotoxins, the infection continues. But if you can nuke those exotoxin bacteria, the infection stops as soon as you kill off the bacteria. So when you're killing the bacteria, it stops making the exotoxins, and the severity of the disease decreases. So your immune reaction helps out. I'm getting at this because we can contrast exotoxins with endotoxins. Now, endotoxins, the easiest way to talk about this is that an endotoxin is essentially a toxic cell wall. So exotoxins are made by the bacteria and squirted out. An endotoxin is where the cell wall of the bacteria itself is toxic. So just the presence of the bacteria. It doesn't have to be making anything. It just has to be chilling inside you. And its presence touching you causes the infection, causes the disease. So that E. coli 0157H7 produces an endotoxin. Its cell wall is toxic. Gonorrhea has a toxic cell wall. Endotoxins generally produce a stronger reaction from the immune system. So endotoxins are going to produce strong fevers, heavy inflammation, lots of immune reaction, maybe immune overreaction even. This is important because we talked about how when your immune system nukes an exotoxin-making bacteria, well, the bacteria stops making exotoxins because it's dead. So the more of them you kill, the less dangerous the disease is. But when your immune system nukes an endotoxic bacteria, the bacteria may be destroyed, but as it decays or if the cell wall is broken apart, the endotoxin is still there. So you're going to keep up with the same severity of infection until the walls are completely digested. So endotoxins produce a longer duration infection. When the immune system gets into it, killing an endotoxic bacteria does not stop the toxin because the toxin was part of the cell wall. So you have to both kill the bacteria and then completely break down the cell wall before the toxin stops affecting you. Whereas with the exotoxin, you kill the bacteria, stops making it. Exotoxins on their own have a short activity duration. So if you're getting a strong fever, you're likely infected with an endotoxic bacteria. So much stronger immune reaction. Since fever is an immune reaction, endotoxic bacteria produces stronger fever, stronger inflammation. So yeah, dangerous. Okay, that's bacteria. Now we're on to viruses. Okay, so viruses are the smallest organism. Remember I talked about how there's debate in the biological community over whether or not viruses are considered alive. I haven't studied viruses a lot, but I've taken virology courses. I've read about them some. And personally, I would consider them a living organism. So I'm going to sort of shorthand call them organisms. Does that mean I am absolutely correct? No, the debate continues, but I think that they're a living organism. You'll find a lot of virologists think they're a living organism. So when I say they're the smallest living organism, remember what I just told you. So the virus is the smallest living organism, right? You thought bacteria were small. Well, you can get large amounts of viruses attacking bacteria. In fact, see this little picture here, right? This is a bacteriophage. I love it because it looks like the frickin' Apollo moon lander, right? There's its little landing pods. These fibers bind to the bacterial cell wall. Here's its little stalk, and here's the little container with its genome inside it. And what happens is these little tails, these legs, bind onto the bacteria, and then they flex, and the whole thing drops down, and this bottom is actually like a needle, and it punctures through the cell wall and squirts its genes into the bacteria. It's metal as hell. All right, so anyway, bacteria are pretty simple. They have a protein coat or shell. That's what we're looking at here in this image and this beautiful artist's rendering. So, and then here's a couple of different images. So they have a protein shell or coat. It's just made up of proteins. Inside the protein coat are enzymes needed to kickstart the bacterial takeover of a cell, and the basic, I just said bacterial takeover of a cell. Enzymes needed to kickstart the viral takeover of the cell and the virus's own genome, right? So some important rules about viruses. They are very specific. A certain virus infects a certain type of cell. So, flu virus affects your digestive system, causes a lot of diarrhea and vomiting. However, it does not cause respiratory symptoms or signs. So, they are specific to the tissue or organs that they infect, or specific type of cell in the case of a bacterial phage. Their whole MO, their mode of working, is to invade the cell. Right? They have multiple different ways to invade cells. Talked about the badass way a bacteriophage invades a cell, but animal viruses, for instance, will hook up with a cell and be like, ooh, it's warm, and they'll bind to the membrane, and different parts of them will bind to the membrane, and it'll sort of be like forcing a blanket around them. They force the membrane around them and then push inside. And then once inside, they open up, pop out their genome, and those enzymes get to work converting the cell into a factory specifically made to manufacture more viruses. That's all that cell does. Once a cell's infected, it's converted into a virus factory. And it makes viruses until it runs out of energy. After that, the viruses are assembled up, they leave the cell, and they infect other cells. They can kill the cell when they leave it. Some viruses don't kill their host cell when they leave it. But either way, the cell is pretty much non-functional. Sucks for the cell. So they invade the cell. They convert it into a factory for making viruses and crank out more viruses and then blow out of that cell to attack all the other cells around it. So really effective at causing infection. So here's a little image of a virus. And this is what I was talking about how it'll pull a blanket around it. So these little blue suction cups are attaching to it. And as they attach, they pull the membrane around. And as the membrane pulls around, it pushes the virus in, pops out its genome, and the cell starts making new virus genomes and new proteins and enzymes. And then the enzymes are assembled inside, and the genome are assembled inside the protein shell. We pop out, and we go and start over again. So it's sort of a cycle of infection. Viruses are pretty scary stuff. That's 36 of 41, folks. Home stretch. Next up is the prion. Since I'm talking about scary stuff, I might as well get into some seriously scary shit. Prions are scary because prion is capable of being produced by your own body. It is scary. So what is a prion? It's just a misfolded protein. So remember, as a protein gets made, we have the primary structure, which is just a chain of amino acids. Then it coils up, or it turns into a little accordion shape, and then it's further compacted into a big ribbon, and then it becomes a functional protein. Well, when you misfold a protein just right, it becomes a prion. A prion is considered an infectious protein. How does that work? Basically, when a prion encounters another protein in the body that could become one, and that protein touches the prion, comes into contact with the prion, it automatically misfolds into a prion. So prions convert normal proteins into prions. This is bad. These misfolded proteins don't dissolve well. And one of the big characteristics of a prion disease is these proteins get into your brain, and then they start to accumulate in your brain, right? So it converts them into prions. They get into your brain, and the misfolded proteins accumulate in your brain. And as you get more and more of them, they start to basically Swiss cheese your brain. So this is a normal brain dense with neurons. Here's Kuru, a prion disease. We see these big white holes in the brain. Creutzfeldt's Jakob disease. Here's big holes in the brain. Scabies, a prion disease of the sheep. Big old holes in the brain. So it Swiss cheeses the brain and causes death. So how bad is a prion infection? If one prion gets into you, you die. It may take a little while for those prions to build up, but there is no cure, no treatment. You will die. So, Creutzfeldt's Jakob disease is a prion disease that's primarily inherited. So there's actually a gene in your body that makes a protein that your body needs to run normally. And it's called a prion protein because that particular protein, if the gene gets mutated just right, turns into a prion. So if that prion protein gene gets screwy, you get Creutzfeldt-Jakob disease, CJD. Now, you might remember good old mad cow disease. Mad cow disease is a prion infection of cows, right? So if a human eats meat from a cow that had mad cow disease and they consume a prion, it causes basically Creutzfeldt-Jakob disease. It's not inherited, but it causes it and kills you. That is why when people find a prion-infected cow, they kill the herd. They don't know how many got infected. So one of the big reasons cows were getting infected is because for a while cattle feed was basically enriched with nutrients by taking waste parts from the cow processing plants, like the brain, grinding it up into the feed. And that gave them a nutritional supplement. But you might remember the brain is where the prions accumulate. So, Scabies is interesting. There's no recorded case of the Scabies prion infecting a human from consuming sheep. So, yay. You can't get Scabies probably. So, yeah, if you get one prion in your bloodstream, you die. What is the way to sterilize meat if there's mad cow disease? No way! If there's a prion in the meat, that meat ain't safe. You can cook it to whatever temperature you like. There's a prion functional in there. They are incredibly durable. You can dump them in acid, and they maintain their structure into levels that, would be destructive to the meat or the person, lethal to the person if you're trying to treat someone that way. That's impossible, though. You inject acid into someone's blood and they die. We talked about that. Anyway, so yeah, you have to basically turn meat into ash to kill a prion. So yeah, I'm scaring you. Okay, let's get out of prions and talk about parasites. 38 to 41, so close. Okay, so what is a parasite? Well, for one, a parasite is a eukaryote. So parasites are eukaryote microbes that cause infections. Sometimes they're not even microscopic. Anyway, there are large numbers of different parasites that can cause parasitic infections. Lots of species. A great example is the malaria parasite. So it's called Plasmodium. 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 The malaria plasmodium gets into your bloodstream, and it kind of rocks it like a virus. It gets into a red blood cell, and it reproduces new plasmodia, and then they pop out of the red blood cell, killing it, and go infect other red blood cells. So it's a red blood cell parasite. What's fascinating is that the malaria parasite has evolved with mosquitoes, such that mosquitoes are what transfer the malaria infection from host to host. They can survive in the gut of a mosquito, and they migrate to the mosquito's salivary glands. So when the mosquito attacks and essentially spits into the next person, because their spit numbs you so that you don't feel the mosquito poking into your blood vessel and drinking. When they spit in there, they spit out the malaria, and then it ravages the person, and another mosquito comes in, drinks the blood, and gets malaria plasmodia, and flies off. They migrate to the salivary gland, and the cycle continues. So that's how malaria do. So, invades the red blood cell, replicates, destroys red blood cells, and is transferred from host to host by mosquitoes. So, it's fascinating. We find malaria anywhere where we find particularly wet, humid, hot climates. Africa is a big hotbed of malaria. We talked about that when we talked about sickle cell anemia. But it used to be that malaria was a huge problem in Florida, actually. So Florida and the Gulf Coast used to have a large incidence of malaria, all the way through to Panama. And so what happened was people got real dedicated with insecticides. In fact, when you see images of old black and white images of trucks driving around just spraying huge billowing clouds of the insecticide DDT, they were primarily shooting for mosquitoes. And so what's happened is that the mosquito that can transmit malaria has largely been murdered off in the Gulf Coast in Florida, and even, the southern Atlantic coast, like North Carolina, South Carolina. Those areas that used to actually have malaria, the mosquito's been wiped out. And that's actually a big goal for treating malaria in Africa, is trying to find ways to wipe out the mosquitoes and find ways to protect people from mosquitoes. There was even this really cool design that people were trying to develop. I have no idea if it ever made it out of the development stage, but it was basically a little mini laser that could, by some characteristic of the mosquito, detect a malaria-carrying mosquito from a normal mosquito and actually like burn it out of the air with the laser. I thought that was awesome. But as we're not talking about sending laser guns to Africa, I don't think that's actually being produced yet. So there's malaria, the Plasmodium parasite that causes malaria. And then there's scabies. Scabies is a good example of a little arthropod, a mite that causes an infection. It's a skin mite. So they're pretty much microscopic, and they burrow into your skin. And once they get deep enough, they eat the matrix of the connective tissue beneath the basement layer. They also eat skin cells and connective tissue cells. They're not going to kill you, but they are going to cause an intensely itchy rash and scaly skin. So they're little skin mites that burrow into your skin. So there we go. There's a scabies mite, and there's an image of the scabies infection. So yeah, skin mites, and they burrow and feed on your skin cells and your connective tissue and the connective tissue matrix. Ugh. Ugh. I pointedly did not put bed bugs in here because holy shit, bed bugs are awful. I do not even want to talk about them. I knew someone with bed bugs. I have like PTSD from dealing with bed bugs. Oh, God. Whoa. Okay. Pathogen transmission. We're going to talk about multiple types of pathogen transmission. There's actually a list of 9 different types. Hopefully, these will be simple enough that we can get through them. Okay, so airborne transmission. What is airborne transmission? Well, when you think of the word airborne, you think, well, it's traveling through the air. That's true, but it's not quite true. So varicella is chickenpox. So you do not have to try and remember varicella. You can just call it chickenpox. And if you do, more power to you if you remember varicella. Anyway, it survives as droplets in the dust. So it has to be transferred out as droplets. So it's in little droplets. Maybe it's saliva. Maybe it's just like a skin secretion, something. But it's in a droplet. And that droplet attaches to dust. And the dust carries it around, is inhaled, and causes the infection once it gets inside with someone through inhaling or it lands on their skin. So it's not airborne in that the pathogen can sort of like fly through the air. It gets into mucus or saliva or spit or something, and that spit, those little droplets end up on dust, and the dust floats around. Direct contact. You get basically a pathogen transmitted through direct contact, requires direct physical contact, right, with the infected person. You have to like rub up against them, or you have to directly inhale their saliva or, sneezes or coughs. You have to directly take in their secretions. So Ebola is a famous direct contact pathogen. During the big Ebola outbreak, there was actually this hilarious song. I don't remember enough of it, but it went, Ebola, don't touch your friends, no kissing, touching. That's all I remember. But basically trying to tell people, do not touch someone who has Ebola, because it is a direct contact transmission pathogen. Ingestion. So, ingestion is how you get a pathogen that goes blah, 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 blah. A pathogen transmitted via ingestion is a pathogen you have to eat. Food poisoning is your common ingestion pathogen. Interestingly enough, polio is transmitted through ingestion. It's not like a direct contact or a sneeze on you, unless someone manages to like sneeze on you and you swallow it. But you have to swallow the polio virus in order to get the infection, which is fascinating. Makes sense. Your skin is a really good barrier. Pathogens generally cannot cross your skin. But if they get into your digestive tract, your mouth, your esophagus, stomach, intestines, a lot easier for a pathogen to get in your system. So, ingestion, eating it, food poisoning, and polio. Migration. This is literally when something goes from a place it should be to a place it shouldn't be. So this is a normal flora thing. Remember how I said some of your skin flora, if they get through a cut, can cause an infection once they get below the skin? That's migration. So migrating from where they should be to where they shouldn't be. It's a normal flora thing. Vertical infection is an interesting one. This is where a mother passes an infection to her fetus or an infection to the infant during birth. So the infection can either pass, it can cross the placenta into the developing fetus. Remember when we were talking about Zika, how it can cause birth defects? Apparently the Zika virus can cross the placenta. So, and then, for instance, gonorrhea will infect the vaginal tract of the mother, so that during birth, the infant passes through the vaginal tract and all the mucus in there and all the gonorrhea. In fact, in the third world, one of the most common causes of blindness is gonorrhea, getting into the infant's eyes during as it passes through the birth canal. So, next up in the pathogen transmission list, which, you know, this is the final thing. 40 is the last informational slide, then we end on a nice, pretty picture. Sexual transmission, right? Had to get here. HIV is our gold standard example of sexually transmitted pathogens. So there's the term STD, sexually transmitted disease, and then there's the term STI, sexually transmitted infection. I keep looking off to the left because my pet praying mantises are like fighting each other and doing weird stuff. They're just babies. Anyway, so sexually transmitted infections are, you know, transmitting the actual microbe, whereas sexually transmitted disease is the disease caused by the microbial infection. Anyway, that's an aside. So sexual transmission. HIV is transmitted via sexual mucus secretions and sperm. So, like, it gets into the ejaculate, and it can get into the mucus lining the vaginal tract. So, gonorrhea, also sexually transmitted. Vehicle transmission. So, think of vehicle, you get in your car, you go someplace else. The vehicle for a pathogen is an object, like a doorknob or a phone or a utensil. One of the most common vehicles for transmission in a hospital is a catheter. So, people will get urinary tract infections, UTI, urinary tract infections, infections of the bladder, the urethra, from a catheter. So that's one of the most common vehicle transmissions is the catheter. But you've also got doorknobs, phones, or eating utensils, or various other things. Any object, really. Second to last is the vector. A vector is different from a vehicle in that it's alive. So vectors are living transmitters, right? Malaria's vector is what now? Come on, say it louder. I can't hear you. Anyway, it's the mosquito. So malaria has vector transmission via the mosquito. Rabies, by the way, is another vector-transmitted pathogen. So the rabies virus is fascinating. It actually has two phases. During the initial phase of rabies, the animal isn't like crazy running around. It sort of stops drinking, has depressed activity, depressed appetite, generally seems sort of down in the dumps. And then, as the animal's infection progresses, the rabies virus migrates to the salivary glands. And then the animal starts salivating heavily. At the same time, the animal gets a fear of water, bodies of water. So it literally won't drink. And that promotes continued salivation and foaming. And this progresses into a phase of the disease called furious rabies. It's literally called furious rabies. And that's what you think about when you think Cujo, running around, snapping, biting. So the rabies virus has a living vector in dogs or raccoons. So not opossums. Opossums don't carry rabies. They're adorable, so don't think bad of them. Okay, so that's vectors, living transmitters. Malaria and mosquitoes, rabies virus, and dogs. Finally, transfusions. So, if you've got a pathogen in your bloodstream and you give blood and then somebody else gets a transfusion, that pathogen gets to them. There are records of HIV being transferred from one person to another via a blood transfusion. So this is one of the things humans work very hard on limiting. There are a lot of tests hospitals run on transfusion blood before they give a blood transfusion. So generally you can be very, very confident about your blood transfusions from a hospital. Now, if you stroll on up to your friendly neighborhood, like street alley pharmacist and be like, hey bro, how about some blood? And get a blood transfusion there, I make no guarantees about your safety. But that scenario, I think, literally has never happened. So, there's the upshot. All right, last picture. Direct contact, either droplets or literally direct contact. Airborne, droplet nuclei, which is their fancy way of saying droplets in dust. Vehicle, objects, like this phone or this cutting board. Vector, like mosquitoes. So, just a nice little picture to drive it home. And holy crap, folks, you made it. Cheers to you. And I'm going to eat my cold dinner.